Pink pigeons. Who are they, where are they, and why do we care? These are all valid questions that any good scientist would start with. You might think pigeon, another rat of the sky. But this is far from the truth. They persist as a conservation miracle. Weighing in at 350 grams and approximately 37 centimeters in length, a healthy individual is remarkably hard to find. Pink pigeons are quite rare and were once almost extinct, with only 10 remaining individuals in 1991. Less than 3 decades later, and this number has increased to 400 due to the beginnings of a captive bred program. Endemic to Mauritius, which is located off the coast of Madagascar and is hardly visible on any given map, 5 locations host wild subpopulations. 4 out of 5 locations are clustered in the Black River Gorges National Park and 1 in the Isle of Le Gret. No demonstrable evidence has shown any genetic differentiations between these areas. Pink pigeons are not naturally migratory and therefore are predicted to have not distributed farther than the island's boundaries due to this. Before genetic comparison studies, the pink pigeon was thought to be a part of the Columba genus, like most other pigeons and doves. However, likely the result of geographic isolation, they are now classified as their own. Not to be confused with their close relative, the Malagasy turtle dove, which also formed a genus. Habitat destruction and introduced predators and disease were once and remain the top threats to continued conservation efforts. One local management implementation has been to include common feeding stations to replace the herbivorous plants once growing on Mauritius. Unfortunately, this introduced congregating individuals to disease. For example, the most well-studied parasite, known as trichomoniasis, affects over half the pink pigeon subpopulations and is detrimental to the functionality of their digestive systems. And even with all these problems, the most well-studied and concerning issue at hand is that due to existing small population size, they struggle with inbreeding depression. Observations of the few hundred individuals indicate that inbreeding has potentially compromised long-term health, like decreases in fertility rates. Recommendations for remedying this low genetic diversity is supplementing the wild population with captive individuals. Arguments for the solution are that distinct or novel alleles have formed, thus providing opportunity for improved environmental responses. But both theory and solution remain to be studied more intensely and have little genetic evidence to back them up. In the long term, we could develop a worse-off population that cannot thrive without reliance on us. We want to ensure that our efforts are not wasted and that future generations can grow to appreciate this amazing species. Pink pigeons are not unlike most other bird species in that they mate for life. And lo and behold, novel alleles may play a role in courtship behaviors and the ability to reproduce at all. If you or individuals cannot find each other, don't find each other suitable in the wild, or are paired incorrectly in captivity, no new birds will be born. Last year, the Bronx Zoo facilities reported that even after a series of eggs were hatched during breeding season, they were all infertile. This should be explained by the fact that female birds commonly produce eggs without a male present because of a seasonal hormonal process. But these females had a male present and the eggs were not fertilized. Therefore, if the males had not found any of the females desirable, we cannot expect any future chicks. Like this figure demonstrates, it takes very little time for either gender to terminate a copulation. But to say a chick were to be born in the future, according to their known life stage timeline, chicks rapidly develop within a couple months and cannot survive without their parents feeding them. So if one parent were to die, this leaves the remaining adult with the heavy responsibility to seek food and protect the chick. In more cases than not, this chick or brood of chicks will not survive with a single parent. Comparing allelic differentiation as well as current age structure and sexing of captive and wild populations is crucial. It would help determine if individuals are maximizing their likelihood of mating and reproducing. To conduct this important work, scientists could collect samples, like blood or feathers, to extract DNA from. They should report molecular sexing because an even sex ratio provides the highest chances of all birds pairing up. But even more so, they need to select and amplify microsatellites associated with mate selection. Introducing more stringent individuals like those in the Bronx Zoo will not necessarily help the wild populations. And if individuals are becoming more or less choosy, we could ask what dynamic reasoning explains this. Perhaps because of captivity, the gene that influences finding a mate regardless of its longevity has been reduced. In many other bird species, like these parrots, an assessment and balance of many different physical factors influence mate pairing. Therefore, enrichment tools like prolonging the time that females interact with males before breeding season to allow them to bond may encourage natural behaviors. Additionally, comparative studies between captive and wild populations need to show that there are not significant differences in genetic structure. A greater disparity between subpopulations and captive to wild populations, indicated by higher FST values, would be detrimental to reproductive viability. This diagram depicts an example relationship between diversity within a group, as shown in the colorful circles, as well as divergence between groups on the dotted lines. The higher FST value between breeding and wild circles means that a speciation event could be occurring. By measuring allelic richness and observed heterozygosity too, it may allude to if there are enough mates to choose from that may also have favorable alleles. If richness is low and females are not laying fertilized eggs, then we could be observing a trend where they don't mate with individuals too closely related to them. This is the unfortunate evidence of the inbreeding depression mentioned before taking its toll. It was inevitable, and we are now just seeing the consequences of perhaps a more homogeneous population reaching the capacity that they're willing to breed. Then it would be recommended to reorganize and share the individuals currently captive around the world to test this theory before placement in the wild. All of the genetic tools and measured parameters that have been mentioned thus far in this presentation are essential because they formulate the basis for a demographic population viability analysis, or a PVA. Due to the lack of data on these factors, a PVA remains to be seen for this species. But a PVA could improve the understanding of resilience and likelihood that any relocation of individuals would be successful due to reproductive trends. Here, nest mortality decreases over time, which is a positive future for this case study. It could be beneficial for evaluating exchange between only captive individuals too. So just to sum up, before even considering future relocation, genetic data influencing mating behaviors, life history traits, and molecular sexing are factors that should be studied as it relates back to inbreeding depression. Additionally, preparation to ensure the longevity of the experiment must be enacted. Firstly, as simulated population growth models have shown, confining the species to a deteriorating region is ineffective, and disease spread must be controlled before allowing individuals to return. If we were to pick relocations now, there must be a surplus of food available and adequate tree space for paired pigeons to mate and nest. Secondly, we could place genetic markers in relocated individuals to determine the success of the initiative over time with occasional capture and release. This is important because more chicks are seemingly indicative of success without the realized understanding of an evolving genetic structure, a genetic structure that by and large changes their attitude for continued breeding. But this isn't to end without hope. Other species like the prairie chicken are a prime example of relocation that went awry before it went right, and it took some trial and error. While pink pigeons may be a more delicate scenario given fewer individuals to start with, we can partially learn from the mistakes made beforehand. And bringing greater awareness to the situation should encourage the institution of new local restrictions on Mauritius and future conservation actions.